Hmm. So, the chemiosmotic theory states that energy derived from electron transfer reactions is temporarily stored as a transmembrane charge and pH differences. Yes, this is correct, because once this gradient that is formed is broken off, when this gradient is broken, then only the energy is actually generated. Okay, so this is correct. Then NADP reduction occurs at the end of the Calvin cycle. No, this is false. Why? Because one in the Calvin cycle, what happens first? The six CO2 molecules are actually assimilated. Okay, then there is formation of ATP, followed by there is reduction of NADP. Okay, so this reduction happens in between the cycle, not at the end, because the last step is the regeneration of the Rubisco molecule. Oh, sorry, ribulose 1,5 biphosphate molecule. So at that point, there is utilization of ATP rather than formation of the NADPH. Okay, so option E is actually false. So the correct answer is option E. Okay, any doubt in this question? Just wait a minute. No, ATP hydrolysis. Yes, ATP hydrolysis means breaking of ATP only. So to release energy. So this was the only question. Yes, in F1 ATP generation happens. That means ATP formation happens. Oh, from that point you are saying, okay. So yes, I also didn't see. So this can not be the yes. Okay, okay. So hydrolysis will not be the case here. This would be ATP formation, not hydrolysis. Sorry, I also missed this thing. Okay, this would be formation. This would be a misprint from my side. Yeah. Thanks for pointing it out. Okay, any more doubts? Okay, so let's move on to the next question. Which of the following are true regarding Rubisco? That is an enzyme used in Calvin cycle. So what can be the correct answer?
Option A, C, E.
A, C, E. Okay. Any more answers? Okay. Okay. Okay, so let's discuss. So the correct answer is A, C, and E. Okay, and here is the explanation. So yes, this is a fact that Rubisco is the most abundant protein in the biosphere. Okay, and then Rubisco, moving to the second point, Rubisco exhibits octahedral coordination with Mg2 plus that is coordinated with six hydrogen atoms. Okay, no. So the Mg2 plus that is present in the Rubisco is actually coordinated with the oxygen atoms. Okay, so it is coordinated with the six oxygen atoms as mentioned in the figure also. Okay, so one oxygen is in the carbamate on lysine 201, two oxygen from the carbonyl groups of glutamate 204 and aspartate 20 sorry, and 2 at C2 and C3 of the substrate. Okay, this is from the substrate C2 and C3, ribulose 1,5 biphosphate, and 1 from the substrate CO2. Okay, so this is the six oxygen atoms that are coordinated with the Mg2 plus of the this thing. Okay, then moving to the third option, Rubisco has a relatively high optimal pH and its activation is regulated by Rubisco activase. Yes, this is correct. So it the optimal pH of Rubisco is relatively high and this is alkaline towards alkaline medium, and its activation is regulated by another enzyme that is Rubisco activase. Okay, then Rubisco activase is a ATP independent enzyme. No, this is a ATP dependent enzyme. Okay, as you can see in this figure also, this is ribulose 1,5 biphosphate. This is lysine 201 of the Rubisco enzyme. Now, first it is unmodified. Okay, then after Rubisco activase comes into picture, it utilizes ATP, releases ribulose 1,5 biphosphate, and is actually this first uncovers the lysine molecule, that is it removes the ribulose 1,5 biphosphate from the active site and then it starts its work. Okay, then Rubisco function in carbohydrate biosynthesis in C3, C4 and CAM pathway. Yeah, CAM plants, yes, this is right. In C3, it's the Calvin cycle, that is the major carbohydrate assimilating pathway. Then C4 is the extension of C3 actually. So in this C4 plants, C3 is the common pathway, and there is another pathway that happens in the bundle sheet cells. Okay. And in CAM plants also, Rubisco is a common enzyme. Okay. Just that the main carbon dioxide assimilation occurs in the night time, not in the daytime in this case. Okay. Then carbamylation of lysine 201 of Rubisco activase initiates the Calvin cycle. So carbamylation of lysine 201 of Rubisco, not Rubisco activase. Okay. So this is also false because this is mentioned as Rubisco activase, not Rubisco. Okay. Lysine 201 carbamylation happens of Rubisco. Okay. That happens that initiates the Calvin cycle. So this is also false. So the correct answer is A, C, and E. Okay. So any doubts in this question? Okay, so let's move on to the next question. Which of the following statements are false? So what can be the correct answer?
Sir, option A, D, E.
A, B, E. Okay.
सर सर बी नहीं, डी।
D. Okay. Okay. Any more answers? Okay, so let's see the answer. So the correct answer is A, B, and E. Okay, and here is the explanation. So first statement is chlorophyll B has a COCH3 group in place of CH3 group in chlorophyll A. Okay, so this is a molecule of chlorophyll and chlorophyll A, and here is the CH3 group in chlorophyll A. And in place of CH3, it's the CHO group in chlorophyll B. Okay, so here it is mentioned COCH3. So this is false. Okay, then B option is the return from the excited state to the ground state during photosynthesis happens via electron transfer. Okay, so this is both electron transfer as well as release of energy. Okay, so this option B is also false because both the thing happens when from the excited state, because this absorbs energy, then only it is moved to the excited state, and once it shifts its electron and release the energy, it comes back to the ground state. Okay. Then difference between the pH of the stroma and the thylakoid lumen is around 3. Yes, this is correct. Then 4 electrons are released when water is split by the oxygen evolving complex. Okay, so here is a mechanism of the oxygen evolving complex. So what happens? Two molecules of water enters this, and here is a complex of manganese. Okay, so manganese actually is a coordination it belongs to the it can easily form the coordination complexes. So it can have a oxidation state of +4. Okay, so what happens? This enzyme, that is P680, this is photosystem 2. Okay, so what happens now? The electron is actually like extracted from this water molecule, and 4 electrons are actually extracted and it enters the photosystem 2 after taking 4 photons. Okay, so and releasing 4H plus into the lumen of the stroma. This releases one molecule of oxygen. Okay, so here 4 electrons are actually being taken from 2 water molecules. Okay, so this is correct. Then magnesium or manganese is central to the oxygen evolving complex. Actually, it's only manganese, not magnesium. Okay, so this is also false. So the false statements are A, B, and E. Okay, So the correct answer is A, B, and D. So any doubts in this question? Yeah, both. It's actually both electron transfer as well as the energy transfer. Okay, so it is both actually, not only the electron transfer. Okay, any doubts in this question? Okay, so let's move to the next question. What are the stoichiometric for the following molecules involved in carbon dioxide assimilation? Okay, and if the stoichiometry of these three compounds are 1, 2, 3, for example, then you have to answer as 1, 2, 3. Okay, so what are the actual stoichiometry of these three compounds that are involved in carbon dioxide assimilation? Okay, so first we have one, two, one. Any more answers? Okay. So before that, what is actually the compound A? So what is compound A? It's actually ribulose 1,5 biphosphate. Yes. Then compound B is 3-phosphoglycerate, and compound C is glyceraldehyde 3-phosphate. Okay. Now in the carbon dioxide assimilation pathway, this is the pathway that happens. So what happens? Three molecules of CO2 comes in the picture, and three molecules of ribulose 1,5 biphosphate attaches with three molecules of carbon dioxide. That forms six molecules of 3-phosphoglycerate. Okay, so three molecules of ribulose 1,5 biphosphate, six molecules of 3-phosphoglycerate, and six molecules of glyceraldehyde 3-phosphate. Okay, so the answer becomes 366. Okay, is the explanation clear for the particular question? Okay, so let's move on to the next question. In stage 3 of the Calvin cycle, where the ribulose 1,5 biphosphate is actually regenerated, so from triose phosphate, so we have to select the correct matches in this context. So what can be the correct answer? Okay, D. Any more answers? Okay. Okay, let's see the answer. So it's A and B both. And first see the process, how it happens. So what happens? One molecule of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate combines, and it in the presence of transaldolase it forms fructose 1,6 biphosphate. Then fructose 1,6 biphosphatase releases its 1-phosphate group and it forms fructose 6-phosphate. In the presence of glyceraldehyde 3-phosphate and the enzyme transketolase, it forms two molecules, erythrose 4-phosphate, that is a 4-carbon sugar, and xylose 5-phosphate, that is a 5-carbon sugar. Okay. Then this erythrose 4-phosphate, in the presence of dihydroxyacetone phosphate, forms pseudoheptulose 1,7 biphosphate. And then xylose by 5 phosphate, in the presence of ribulose 5 phosphate epimerase, it forms ribulose 5 phosphate. Okay, these both are 7 and 5 carbon compounds respectively. Then pseudoheptulose 1,7 biphosphate forms pseudoheptulose 7 phosphate, releasing its 1 phosphate group, and ribulose 5 phosphate actually converts to ribulose 1,5 biphosphate, and this is the first molecule of ribulose 1,5 biphosphate that is formed. Okay, then uh, from this pseudoheptulose 7 phosphate, it combines with glyceraldehyde 3 phosphate in the presence of the enzyme transketolase. It forms ribose 5 phosphate and xylulose 5 phosphate. Okay, and this xylulose 5 phosphate again transforms to ribulose 5 phosphate, then to ribulose 1,5 biphosphate, forming the second molecule. Okay, then this ribose 5-phosphate is actually converted to ribulose 5-phosphate, okay, by the isomerase enzyme, and this ribulose 5-phosphate is converted to ribulose biphosphate. This is the third molecule. Okay, now let's see the question. So we have fructose 1,6 biphosphate and the enzyme trans aldolase. So fructose 1,6 biphosphate is here, and this is the glyceraldehyde molecule, and this is dihydroxyacetone phosphate. Okay, so in the presence of sorry, yes, so glyceraldehyde 3 phosphate in the presence of enzyme transhaldolase, it can convert to fructose 1,6 biphosphate. Sorry. Okay, okay, wait. So this is 3b and 1. Okay. Yes. So from glyceraldehyde 3-phosphate, in the presence of the enzyme transketolase, xylulose 5-phosphate is formed. Okay, so this is the first option. So xylulose 5-phosphate, transketolase, and glyceraldehyde 3-phosphate. So this is the first option. Then the second one is 1A and 3. That means fructose 1,6 biphosphate, trans-aldolase, and dihydroxyacetone phosphate. So yes, dihydroxyacetone phosphate in the presence of trans-aldolase, it forms fructose 1,6 biphosphate. Okay, so this option B is also correct. Okay, then we have 2A and 1, that is pseudoheptulose 1,7 biphosphate, trans-aldolase, and glyceraldehyde 3 phosphate. So instead of glyceraldehyde 3 phosphate, it must be dihydroxy acetone phosphate. Okay, so that's why option C is false. Then 3B and C, that is xylulose 5 phosphate, trans ketolase, and then dihydroxyacetone phosphate. No, this would be glyceraldehyde 3 phosphate like A, so this is also false. Then we have sedohoptulose 7,7 biphosphate, trans aldolase, and erythrose 4 phosphate. Yes, this is also correct. Okay, because erythrose 4-phosphate in the presence of trans-aldolase, we can form pseudoheptulose 1,7 biphosphate. Okay, so this option is also correct. Option E is also correct. Okay, so here option E will also be added. Okay, because we have, oh sorry, this would be trans-ketolase, so this is false. This would be A instead. Okay, so option A and B will also only be correct. Okay, so any doubts in this question? Okay, let's move on to the next question. Which of the following enzymes associated with the Calvin cycle require light for their activation? So, what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option E. It would be option E. Okay, because all of these enzymes are actually being involved in the assimilation of carbon dioxide. Okay, so that's why these enzymes would require light for their activation. So it is option E. Okay, so let's move on to the next question. Which of the following statements are true regarding oxygenic photosynthesis? So what can be the correct answer?
Sir, option C and D.
C and D. Okay. Any more answers? Okay, so let's see the answer. So it's A, C, and D. Okay, so the plasma membrane of Helicobacter selenium contains patches of light-absorbing pigment bacteriorhodopsin. Okay, so yes, this is correct. So what happens in the membrane of the this bacterium? The bacteriorhodopsin is actually present in patches like this. So this helps in absorbing the light and formation of the ATP. Okay, so this actually is present in the this bacterium Helicobacter selenium. Okay, this is correct. The ATP synthesis in bacteria, cyanobacteria, mitochondria, and chloroplasts do not share a common evolutionary precursor. No, this is false. They have some evolutionary common precursor. That's why the basic structure of ATP synthesis is same. Okay, so that's why this is false. Many photosynthetic or microorganisms obtain electrons from photosynthesis from donors such as H2S and acetate. Yes, this is correct. Then there is a functional homology between cyanobacterial cytochrome C6 and the plant plastocyanin. Yes, this is correct. Okay. Then we have modern purple bacteria have a PS1 like electron path, and the green sulfur bacteria have a PS2 like path. So this is the cycle for the purple bacteria, in which there is pheophytin quinone reaction center, that is type 2 type of reaction center. Okay, not type 1. So this option is also false. And in the green sulfur bacteria, it's the FES reaction center, that is the type 1 reaction center. So it can actually activate this NAD reductase. Okay. So hence this E option is false. Okay. So the correct answer is option A, C, and D. Okay. So any doubts in this question? B and A. Okay. So in case of Helicobacter selenium, what happens, because this is a bacteria, okay, so it doesn't have any renowned organelles present in that. So what happens, in the membrane of this bacteria there are patches of the bacterial rhodopsin that is present. Okay, so these patches actually help in absorbing the light and formation of the energy. Okay, so this happens in the cell membrane of this bacteria. Unlike plants, we have in plants we have a specialized structure called as chloroplast. In chloroplast we have sorry, just a minute. So in plants we have a specialized structure, that is chloroplast. And in chloroplast, what happens? There are specific structures, that is uh, stroma, so in which the thylakoids are maintained. Okay, so in these thylakoids, the chlorophyll molecule is actually present. Okay, so this is the first point. And then ATP synthesis in bacteria, cyanobacteria, mitochondria, and chloroplast do not share a common evolutionary precursor. No. Why? Because in all the cases, being it mitochondria, chloroplast, or bacteria and cyanobacteria, the basic structure of ATP synthesis remains the same. Okay, it both have F0 and F1 parts, and F0 is involved in the breaking of the proton gradient, and F1 is involved in the formation of ATP. Okay, so that's why they share a common evolutionary precursor. That's why the structure of all the four ATP synthesis is nearly the same. Okay, so that's why this statement is false. Okay, is the explanation fine now? Okay, so let's move on to the next question. Which of the following statements are false?
Sir, option A, B, D.
Okay, A, B, and D. Okay, B. Okay, okay. So let's see the answer. So it's A, B, and D. Okay, so in cyclic electron flow involves PS1 and PS2. So no. In cyclic flow, what happens? Only PS1, only PS1 and cytochrome B6F is involved. Okay, not PS2. So this option is false. The reaction centers of PS1 and PS2 are P680 and P700 respectively. No, it's the reverse. For PS1 it's P700 and for 2 it's P680. So this is also false. Cyclic photophosphorylation is the process of proton pumping by the cytochrome B6F complex and the phosphorylation of ADP to ATP. Yes, this is correct. Okay, because the cyclic photophosphorylation occurs from here, and here the electrons are transferred from the cytochrome B6F to plastocyanin, then to P700, followed by its activation to the NAD reductase complex. So here, because of the proton gradient formation, ATP is also formed. Okay, so this is correct. Then carbon assimilation reactions require ATP and NADPH in the ratio 2 is to 3. No, this is false. Okay, because the ATP is 6 and NADH is also 6, and here 3 ATP are required for the regeneration. So it would be 3 is to 6 or 1 is to 2 rather than 2 is to 3. Okay, so this is false. Then PS2 is located almost exclusively in the tightly appressed membrane stacks of the thylakoid grana. Yes, this is correct. Okay, So the incorrect statements out of these are A, B, and D. Okay. So any doubts in this question? Okay, so let's move on to the next question. Which of the following statements are true with respect to cytochrome B6F? So what can be the correct answer?
Sir, true statement is A, B, C, E.
A, B, C, E. Okay. Okay. So the correct answer is A, C, and E. Okay. So now let's see. The cytochrome B6F complex contains a beta cytochrome with two heme groups, a Reiske iron sulfur protein, and cytochrome F. Okay. So in the structure, it's shown it's cytochrome F, cytochrome B6. Okay. Then we have a Reiske iron sulfur protein and two heme groups. Okay, that are attached to one here cytochrome B and other in the cytochrome F. Okay, so this is correct. Then we have cytochrome B6F conveys electrons from a reduced quinone to a water soluble protein that carries two electrons. Okay, so it is not a water soluble protein. Okay, so cytochrome B6F conveys electrons from a reduced quinone. So this is actually a reduced quinone. Okay, to a water soluble protein that carries two electrons, and it carries the electrons from the cytochrome B6. Okay, so cytochrome B6 is not a water soluble protein. Okay, so that's why it is false. Then Q cycle occurs within this complex, yes, because the quinone, after reduction, it transports its electrons to cytochrome B6 that helps in transferring the electrons. So yes, this is correct. C. And then in chloroplast, the direction of proton movement is from the thylakoid lumen to the stromal compartment. Okay, let's see. No, it's the other side. It's from the stroma to the lumen. Okay, so it's not the other way around. So this is false. The measured difference in between the stroma and the thylakoid lumen represents a thousand-fold difference in proton concentration. Yes, this is correct because the pH of the stroma is pH 8 and that of the thylakoid lumen is pH 5. Okay, because here it's the accumulation of the protons and from here. So because that's why pH is less than uh, is less in lumen as compared to the stroma. Okay, and this difference is 1,000 times. So yes, this is correct. So the correct statements are A, C, and E. Okay, so is the explanation fine? Okay, so let's move on to the next question. The Z scheme outlines the pathway of electron flow between the two photosystems and the energy relationships in the light reaction. What is the stoichiometry of photons for the following Z scheme equation? So what can be the correct answer? Okay, okay. So the correct answer is 8. This is the final reaction of the photosynthesis that occurs. So we have 2H2O that is involved in the production of one oxygen molecule. Two NADP molecule form two NADPH. 8 photons are required for the activation and 2 H plus are released from two water molecules. Okay, so this is 8 photons only. Okay, and how 8 photons? 4 for PS2 and 4 for PS1. Okay, so 4 photons are required for the activation of PS2 and 4 photons are required for the activation of PS1. Okay, is the explanation clear? Okay, so let's move on to the next question. Which of the following statements are true? So, what can be the correct answer? A, C also. Okay, A and C. Okay, the correct answer is A and C. Now let's see the options. So xylulose and ribulose are epimers. Okay, so this is the structure of xylulose and this is for ribulose. Okay, so both are 5 carbon, and at this third carbon, the there is shift in the position of OH and H. Okay, so that's why these are the epimer molecules. Now, there is only one way to get to ribulose 1,5 biphosphate from glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. No, this is not correct. Okay, because there can be multiple ways from which we can get the molecule ribulose 1,5 biphosphate by combining different carbon compounds. Okay, so this is false. Then TPP is a cofactor for the reaction between fructose 6 phosphate and the glyceraldehyde 3 phosphate. So yes, this is correct. Okay, then we have both ribose and ribulose are ketone sugars. No, ribose is an aldose sugar because it has aldehyde group. Ribulose is a ketone sugar. Okay, so this is also false. So the correct answers are A and C. Okay, so any doubt? Okay, so let's move on to the next question. During photosynthesis, that how many photons must be absorbed for every oxygen molecules produced? Just now we discussed this question. Yes, the correct answer is 8. Just now we discussed for 2 molecules of H2O, 2 molecules of NADP, and 8 photons, we get 1 oxygen molecule, 2 molecules of NADPH, and 2 H plus H. Okay, so this is 8 photons. Now let's move on to the next question. The oxygen splitting complex releases how many protons? So the correct answer is option 4. Okay, just wait a minute. Yes. So the answer is 4 protons, and here is the explanation. So from the oxygen evolving complex, what happens? Two molecules of water comes in, and 4 protons are actually taken in a stepwise manner into the stroma. Okay, sorry, into the lumen. So let's move on to the next question. Kelvin cycle consumes how many molecules of carbon dioxide? Okay, any more answers? Okay, so the correct answer is 3, and here is the explanation. So, in the Calvin cycle, only 3 molecules are actually assimilated by 1 molecule of Rubisco. Okay, and 3 molecules of ribulose 1,5 biphosphate are actually involved. So, a total of 3 molecules of carbon dioxide are first consumed in a single Calvin cycle, not 6. Okay, is the explanation clear? Okay, so let's move on to the next question. Each complex in PS1 contains how many different proteins? Okay, so the correct answer is 11, and here is the explanation. So, PS1 has this particular type of structure, and this is associated with 11 different proteins. Okay, so it is composed of 11 different proteins that forms a complex, and it also contains these molecules that are involved in the electron transfer. Okay, so this is 11 different proteins. Okay, okay, so let's move on to the next question. Which photosystem reaction center requires greater energy to excite? Okay, so both said A. So yes, the correct answer is A. Okay, and the reason is because 680 is the wavelength that it requires for the activation. Okay, and energy is inversely proportional to wavelength. Every time you remember this. So whenever the wavelength is less, that means energy is high. So this P680, that means PS2, actually requires greater energy to excite rather than PS1. Okay. So the correct answer is option A. Is the explanation clear? Okay. So let's move on to the next question. Identify the molecule in the following figure. Okay. So the correct answer is option C. It's bacteriochlorophyll A. Okay, and okay, so this is a different, nearly the same structure. Okay, just here it's CH3C double bond O. Okay, here. So that's why it makes it a different structure as compared to chlorophyll A. Okay, so this is a bacteriochlorophyll A molecule here represented. Okay, so let's move on to the next question. One strategy to increase the flux through a metabolic pathway is to engineer the slowest enzyme in that pathway and increase its reaction rate. In case of the Calvin cycle, which enzyme would be an ideal candidate for this kind of engineering? So, what can be the correct answer? Okay, okay. So, the correct answer is option A, that is Rubisco only, because Rubisco is the slowest enzyme. Okay, so this is the slowest enzyme of the pathway and its turnover number is also very less. Okay, so we have to engineer this enzyme so that we can increase its activity through a metabolic pathway and increase its reaction rate also. Okay, so the correct answer here is Rubisco. Okay, so let's move on to the next question. Which of the following molecules would could organisms use as a reducing agent from which to extract the electrons? So what can be the correct answer? Okay, any more answers? B. Okay. How can you use oxygen for reduction? Okay. So the correct answer is A and C. Okay. So both water and hydrogen sulfide are actually used as reducing agents, and we can extract electrons from them also. Okay. So water is used as a reducing agent in higher plants that helps to release the oxygen, and hydrogen sulfide is actually used in lower bacteria. Okay, so from that it actually utilizes this as a electron source. Okay, carbon dioxide and oxygen both are oxidizing agents. Okay, so we cannot use them for extracting any kind of electrons. Okay, is this fine? Okay, so let's move on to the next question. In plants with certain mutation, radioactive labeling experiments were used to show that 3-phosphoglycerate was being converted to dihydroxyacetone phosphate in the stroma. However, this dihydroxyacetone phosphate was not contributing to the production of sugars in the cytosol. Which of the following proteins might be defective due to this mutation? So, what can be the correct answer?
Sir, correct option is B.
B. Okay, let's see. Any more answers? Okay, so what happens when this Calvin cycle is running in the stroma? So this CO2 molecules assimilated with RUBP, that is ribulose 1,5 biphosphate, and it forms 3-phosphoglycerate followed by 3-phosphoglyceraldehyde. Okay, now this 3-phosphoglyceraldehyde is converted to DHAP, which then transported to the cytosol. Okay, so this is stroma and this is cytosol, and in the cytosol this DHAP is involved in the formation of sucrose. Okay, the simpler pathway. Now what happens, it is actually 3-phosphoglycerate was being converted to dihydroxyacetone phosphate in the stroma. However, this DHAP was not contributing to the production of sugars in the cytosol. So it is present in the stroma and it's working fine, but it's not working fine in the cytosol. So what will happen? There can be a mutation in the enzyme that is involved in the transportation of the DHAP. Okay, because with the limited information that we have seen in the question, there is nothing mentioned, just that the particular compound is not working in the cytosol, but it's working fine in the stroma. So that means the transporter is actually mutated here, not anything else. If any other more information would have been provided with respect to the number of enzymes involved or anything else, then we would have commented on other things. So here, the antiporter, that is pi triose phosphate antiporter, that involves in the transport of the DHAP from the stroma to the cytosol is mutated. That's why DHAP is not able to go in the cytosol itself. Okay, so the correct answer is option C. So, any doubts in this question? Okay, so the next question is the enzyme catalyzing which of the following Calvin cycle reactions will be affected in case of vitamin B1 deficiency? Okay, B. Then any more answers? Okay, so the correct answer is option A and D. Okay, so vitamin B1 is basically thymine pyrophosphate. Okay, so it is involved in two reactions in the regeneration of ribulose 1,5 biphosphate. First is the where xylulose 5 phosphate and glyceraldehyde 3 phosphate, glyceraldehyde 3 phosphate and ribose 5 phosphate are reacted and xylulose 5 phosphate is formed. Okay, and in the second reaction in which the pseudoheptulose 7 phosphate is actually converted to ribose 5 phosphate. Okay, so option A is erythrose 4 phosphate and xylulose 5 phosphate from fructose 6 phosphate and glyceraldehyde 3 phosphate. So yes, this is correct. Then from xylulose 5 phosphate and ribose 5 phosphate from glyceraldehyde 3 phosphate and pseudoheptulose 7 phosphate. So this is also correct. Okay, so this is basically a that is TPP is basically involved in trans ketolase enzyme. Okay, that's why these two options A and D are correct. Okay, is the explanation fine? Okay, so let's move on to the next question. Which of the following cytochromes are directly responsible for proton pumping? So, what can be the correct answer? Okay, so the correct answer is option A and D. Okay, so cytochrome BC1 complex is present in mitochondria, and it actually helps in transferring the hydrogen ions from the matrix to the intermembrane space. Okay, and cytochrome B6F complex, just now we studied that it's present in the stromal membrane, thylakoid membrane. So, and it's basically, uh, sorry, thylakoid membrane, and it also helps in shifting the hydrogen ions from the stroma to the lumen. Okay, so these two cytochromes are helps in proton pumping. Okay, so any doubts in this question? Okay, so if not, then we'll close the session for today. So if you guys have any questions regarding the presentation or the week 9, you can ask. Okay, if not, then we'll close the session for today. Thank you guys for joining in. Thank you.